Hi, listeners. Welcome to episode 98 of Cell and Gene, the podcast. I'm your host, Erin Harris. So, for this one, I'm talking to Dr. Simrit Parmar, founder of Cellenkos, and the director of therapeutic manufacturing and entrepreneurship at Texas A&M University. Cellenkos is a clinical stage biotech company developing umbilical cord blood-derived T regulatory, or Treg, cell therapies for autoimmune and inflammatory diseases. Simrit developed a cord blood-derived regulatory T-cell therapy platform in her research laboratories while a tenured associate professor at MD Anderson Cancer Center in Houston, Texas. Listeners, trust me, she is remarkable. Simrit and I talk through the vision behind Cellenkos, the advantages of cord blood-derived Tregs over other sources, and how their Crane technology platform enhances precision in targeting diseases. I really enjoy talking about scalability, and she and I talk through the challenges of scaling up off-the-shelf Treg therapies. This is a really informative episode, and I appreciate you for giving it a listen. Here's my discussion with Dr. Simrit Parmar, founder of Cellenkos. Enjoy. Simrit, hi. Welcome to Cell and Gene, the podcast. Thanks for your time.
Thank you for having me.
Absolutely. So tell us a little bit about Cellenkos and why you founded it.
So, Cellenkos is a clinical stage company. We strive to deliver life-changing regulatory T-cell therapy treatments to patients with autoimmune diseases, inflammatory disorders. And these are truly off-the-shelf therapies that can be cryopreserved, stored for years, can be administered in the doctor's office in the outpatient setting through a peripheral IV line. And more importantly, Cellenkos' Treg cells can seek out the diseased areas, resolve the inflammatory processes, and restore homeostasis and normalcy. Why I founded it goes back several years. The technology was actually founded in my laboratory at MD Anderson Cancer Center. And at the time, I was working as a physician scientist with focus on treating graft versus host disease, also called GVHD. And it is a fatal complication of life-saving allogeneic stem cell transplant for patients with leukemia, with lymphoma, with myeloma, and many other malignancies. So as a transplant physician myself, I was haunted by the fact that we were able to cure patients with the deadly disease only to lose them to GVHD. So I joined this quest to find novel treatments to protect our patients, to cure this terrible, terrible complication. And the findings in my laboratory were actually quite promising. They were translated into phase 1-2 trial where we saw encouraging results. And it was in year 2016 that my invention was spun out into a biotech company, which now is Cellenkos. One of the very first decisions, and I wanted to emphasize on that as our story, was that as a small biotech with limited funding, we made an early decision that we're going to establish our own manufacturing process, and we will control our supply chain, we'll control our storage and distribution. And I think that insight came from the fact that I was so much in the ethos of stem cell transplantation, and I actually knew what it takes to develop a cell and gene therapy therapeutic. So what we did was we established this specific GMP facility. And as a result, we were able to establish our proprietary Crane process. We were able to create a pipeline of 4 distinct differentiated products. And as I speak to you today, our company has 5 company-owned, fully owned INDs. We have completed 4 phase 1 trials in different clinical indications, more than 75-something patients. And proudly, we have also finished a multi-center randomized placebo-controlled trial. So that's us.
Well, very, very exciting and certainly very interesting, and which we'll get to a lot of what you discussed as we go through our discussion today. First, I want to talk a little bit about, from an umbilical cord blood-derived Treg cells, compared to other sources. What advantages do they have, particularly in terms of, the lack of HLA matching requirements?
A great question. So Isolating T regulatory cells from umbilical cord made sense. And before I go into all that, I want to tell you the story of Treg cells or T regulatory cells. So all of us have Treg cells in our body. Every day, our body is exposed to different attacks, whether it is antigens from outside or bacteria, inflammatory processes, or our own T cells in our body that can sometimes attack our own cells and create autoimmunity. The Treg cells are present in our body. These are part of our immune system. They constitute less than 5% of our cells, and yet able to block off and evade all these continued millions of attacks every day. So these are very special cells. These are intelligent cells. These are cells that are able to assess the situation and then take an action or resolve inflammation based upon what is the intensity of attack. So going back to why umbilical cord blood, I want you to think about it that a mother has a baby and the baby is 50% mismatched to the mother, but still the nature allows it. And the cord blood that connects, or the cord blood or the cord that connects the baby to the mother consists of these cord blood, and it consists of many immune cells that are part of the machinery that protects the baby from any kind of immune action or adverse immune action from the mother to the baby. So we thought that the nature has bestowed very strong capabilities on this subset of cells, and why not take this subset of cells and create a therapeutic out of it. Not us, but for the past several, maybe 2 or 3 decades, there has been ample evidence in the field of allogeneic transplant that when you use cord blood as a source for stem cells, they are permissive. They can be given across HLA barrier. So we basically derived from that experience and translated that into our product, that if our product is derived from the umbilical cord blood, which are naive cells, then they should be able to evade the immune barrier. It can be given across HLA barriers. So now what we have done is the very first product that we gave to the patients, it was minimally matched, and we gave a single infusion, one and done, which is CK-0801. And Erin, that was a fail-fast strategy because we wanted to see that what we are developing, does it actually work? We gave no immune suppression, no additional immune suppression. We gave no lymphodepletion, no chemotherapy. Single infusion, one and done. And what I can share with you is that not only our cells were received very well, they were given through a peripheral IV, but the patients showed amazing results. We published these data in New England Journal of Medicine Evidence. Dr. Kadia was the first author, who's also the PI of the trial. And what it showed was that single infusion of Cellenkos' cord blood Tregs, allogeneic Tregs, called CK-0801, were able to induce independence from blood and platelet transfusion that lasted almost 2 to 3 years. So such is the efficacy. And last but not the least, you have to see that these Treg cells, they implement multiple mechanisms of action that are actually HLA independent. They go to the source of the inflammation, which is the antigen-presenting cell. They engage with it with, cell surface receptors, cell ligand. They can deplete the cytokine, which is IL-2, survival cytokine, and they secrete suppressor cytokine called IL-10. And last but not the least, they are then able to neutralize or the cytotoxic T cells that are continually driving the autoimmune process. So because of this multi-pronged approach, which is HLA agnostic, we were able to create a whole pipeline of therapeutics that are that are derived from the umbilical cord blood. And once we are able to productize it, we are seeing these incredible outcomes.
Good. Thank you for that detailed description. That's helpful. I want to talk a little bit about Cellenkos' Crane technology platform. So how would you say it specifically perhaps differentiates cord blood-derived Treg cells for targeting different diseases?
So Crane is a very interesting acronym that we developed to name our proprietary technology, which is cord blood regulatory T cells that are activated and enriched. And there are two main ways of creating these differentiated products. So one is enrichment of existing cell surface markers on the Treg cells. And the other one is expression of new antigens, creating genetic modifications, including lentivirus, CRISPR, etc. The current products that are in clinical trials belong to the first category, where we have been able to enrich the existing cell surface receptors. And the reason for that, why did we do that? Why not just use naive or undifferentiated cell? Is because the theory that we have, and now it has been validated by many, many other scientists, is the homing and trafficking of Treg cells and any other stem cell is extremely important, because if you're able to deliver a homogeneous cell population at the same time to the area of inflammation, you can have a much better resolution compared to if they are trickling one by one. So what we thought was that if we are able to leverage this lock and key phenomena, where the lock is the ligand which is expressed in the target tissue, and the key is the antigen that is present on the Treg cells. By creating this affinity mechanics and by leveraging, which is already existing in terms of homing and trafficking, we developed this technology and we invented these new methodologies where we were able to create cells that are enriched in specific cell surface receptors. And I would like to give you an example, which is amazing, is our product called CK0804. And CK0804 is a product that we have developed for treating patients with myelofibrosis. On its cell surface, we enrich the Treg cells on CXCR4. So naturally occurring Treg cells also express CXCR4, expanded Tregs also express CXCR4. But what we did by our enrichment process and enrichment step was to create a new cell population that doesn't exist in nature, which has a very high expression, CXCR4 high. It is homogeneous. They have different epigenetic signature. They have different homing capabilities, and they have different phenotype and ultimately functionality. The results of our preclinical data was published by Dr. Huang in iScience Cell Press. And the clinical data is also very encouraging. So going back to your question, that what makes it so different is that we are recreating a cell identity, we are recreating a cell transit mechanics, and we are preserving the cellular intelligence that I initially expressed, that these are very intelligent cells. We don't disturb that function. It's almost like a GPS. We give them a navigation, we give them a map, and we pre-program the GPS as to which tissue they have to go to, so that we are able to deliver the payload in an abbreviated period of time and create quantified mechanics around it.
Excellent. Okay. We're going to get even more into CK0804 in just a little bit. But before we do, I do want to talk a little bit about safety profile of your Treg cell therapies. How does the safety profile of Cellenkos' Treg cell therapies, how do they compare to other cell therapy approaches?
Great question, Erin. So I will tell you there are three main ways or three main areas in which our cells, specifically now we are talking about safety, that differentiates it from any other cell therapy, whether it is Treg or other cells. So one is patient-centric. Patient-centric means that we don't expose the patients to any additional processes. So most of the cell therapeutics that are out there are autologous. That means you have to take the cells from the patient themselves. And what it involves, Erin, is to put the patients on this machinery or apheresis process where there is a shift in the cell, in the volume of the patients. So liters of fluids have to go out of the body. The machine has to select the white cells and put rest of the cells back in. And it portends like a central line insertion. It's a lot of stress on the patient. There are a lot of complications of that can happen. In my own career, I have seen patients who have gotten, thrombosis, they have gotten low blood pressure. So we completely isolate, or we completely avoid these kind of mechanics that can expose the patients to any kind of risk. So our cells are completely off the shelf. When babies are born, these cord blood units or cord blood is actually donated to public cord blood banks, which are already saving these cells through very much validated as well as standardized methodologies, which are overseen by the FDA. So our starting material is extremely safe. It has a great it has a great profile and storage signature that is validated. So as a drug developer, we use cord blood units simply as a reagent. And this reagent has already done its full profiling so that none of these are a surprise. So that's number one. Number two is a lack of giving any kind of additional chemotherapies, which is goes to the product-centric. So our cells do not require for the doctor or the patient to receive any kind of chemotherapy, lymphodepletion. So traditionally, the CAR T cells that are out there, they require additional chemotherapy like fludarabine, cyclophosphamide, certain allogeneic products require Campath, which is a huge immunosuppressor agent. We don't need any such thing. It's simply patients are coming in, they get the cells and they go home. So that's number two, that we are not prescribing any new treatment to be given as an adjunct to our cells, because our therapeutics stand alone. And last but not the least is that the ability to give or the administration of the cells, our patients are treated in the outpatient setting through a peripheral IV. We don't need any central IV requirement, and they can go home the same day. The reason I can tell you about these three ways that our cells are differentiated from other cell and gene therapies is based upon our clinical data. We have, like I said, treated more than 75 patients across different clinical indications, including during COVID. In a placebo-controlled trial, our cells were given to very sick patients who are already on vasopressors, who are mechanically intubated, and we didn't see any difference, any safety issues compared to the placebo. The only observation that we have is that sometimes patients who have sulfur drug allergies can react to the excipient that we use to freeze the cells, which is called as DMSO, and that has been described very well in the literature. So all in all, our cells by themselves have a really good safety profile. These are well tolerated by the patients. These don't require patients to undergo any onerous procedures, and they can be given intravenously as an outpatient. So I think that we're very excited that we can bring these kind of therapeutics, especially in patients with autoimmune diseases. These are not deadly diseases. These are not fatal diseases. So exposing patients to chemotherapeutic drugs, I feel, is too much. So we feel that our cells can fill that niche.
Good. That's great. I want to go back a little bit and talk about, bring back up CK0804. Share with us a little bit about the phase 1b data that you shared and presented at ASH 2024, so last year, but particularly regarding efficacy signals and safety.
Thank you. So CK0804 was presented by Dr. Masarova at ASH conference, the American Society of Hematology, last year as an oral presentation. And I think the reason our abstract was chosen for such a great audience is not only because of the brilliant efficacy, but also the great safety profile. And as you know, patients with myelofibrosis are very sick. They are miserable. They have big spleens. They have sometimes requiring transfusions. They have symptoms of fatigue, of just not feeling well. And a lot of therapeutics, there is, great advance in this field, but they come with their own bag of side effects. And that's really limiting some of the implementation of novel drugs. Where our drug, CK0804, differentiates itself is that our patients are able to tolerate these drugs very well without any side effects. Number two is that they start feeling relief in their symptoms almost immediately. And these are anecdotes, these are reports that the patients have communicated to their treating physicians. The most significant data points that I can share with you is that out of the 9 patients who were treated in the phase 1 safety run-in, all 9 patients reported an improvement in their symptom burden. And specifically, 6 out of 9 patients had more than 50% reduction in their symptom burden. When we dissected what type of symptoms were getting better, the 2 main symptoms that stood out was fatigue and early satiety. And as you know, fatigue is a really troublesome symptom subset, which is plaguing these patients because of not only the burden of spleen enlargement, but also the systemic inflammation that these patients suffer from, of the anemia that they are also exhibit. Number 2 is that early satiety is also representative of the splenomegaly. So the fact that we were seeing specifically reduction in these 2 parameters gave us a lot of optimism. The second important observation was decrease in the spleen volume. So Erin, I want to remind you that these patients had already failed 3-4 lines of therapy. There's one patient who is 80 years old. She's failed like 6 lines of treatment. She's been suffering from myelofibrosis, and initially she had polycythemia vera for almost 4 decades. Oh, wow. So this specific woman that I'm talking about, she also had transfusion requirements. She has completed her treatment. She is a year out from her last infusion, and her quality of life has improved dramatically. She is not requiring transfusions. She feels better. And this is really the longevity of the response, or the stability of the response, that we are very excited about. The spleen volume, the spleen burden I want to mention is because it's a organ where, what we call is extramedullary hematopoiesis. That means when the bone marrow shuts down because there is so much of fibrosis, the spleen takes over. And therefore, it starts doing the job of the bone marrow. So these spleens are large, they are painful, they are stretching the capsule, and people are miserable. So in our patients, out of the 6 evaluable patients, 4 had a decrease in their spleen volumes, and 2 had a decrease that was more than 25%. So this relief in the spleen volume was really remarkable. And last but not the least, we saw significant decrease in inflammatory cytokines, including TGF-beta. So what we feel is that by decreasing the symptom burden, improvement in the transfusion response or transfusion burden, decrease in the spleen volumes, and decrease in the inflammation, it ties the story together. And it's all in linearity to when the cell infusions were done. Because these 9 patients did so well, we have now opened the expansion cohort where we are treating patients with a more intensive dosing with 4 weekly inductions followed by 5 monthly infusions. So we are very excited, and hopefully we'll share our new data as soon as it comes out. But
absolutely, we'll be following to me, so that when it is available, we would love to, discuss it on Cell and Gene for sure.
Thank you.
I do want to talk a little bit about the challenges in scaling up production of, like we've been talking about, the off-the-shelf Treg therapies for larger clinical trials and potential commercialization. When it comes to true scaling up, what are the challenges?
So, in the scaling up, I think there are 3 main challenges. 1 is stability of the cell, 2 is the purity of the cell, and lastly is the number. So because our cells are derived from the umbilical cord, we have checked box number 1. So if you look at the Treg cell therapy companies that are out there, especially the ones that take the cell from an adult, or we call as autologous Treg, they are plagued by the fact that these cells can flip-flop. And what I mean by that is that the Treg cells, sometimes when they are derived from the adult, can transform into an effector T cell. And by doing so, it leads to instead of calming the inflammation, aggravate the inflammation. So that's a problem. And our cells don't have it because they don't carry the gene ROR gamma T, which is responsible for transforming these cells into those Th17 type bad cells. Number two is the purity. So we go to extra lengths to make sure that the starting material is of certain proprietary characteristics when we select the cord blood unit. Secondarily, when we isolate the Treg cell, that's a very critical step. And we have extensive phenotype analysis that allows for us to have a go, no-go decision making. And last but not the least is the expansion and storage. So right now we have spent enormous amount of resources to automate the expansion process. And it's important, Erin, because when we are expanding these cells to, let's say, from 20 million cells to 10 billion cells, it's a huge scale. And in order to make sure that we capture the not only the cell volume, but the cell number and don't compromise either during the freezing or during the harvesting. So all those processes need to be automated so that we take out human error out of this whole expansion cohort. So we have in our company specifically, we have now been able to overcome those challenges. And an important aspect is the quality of the quality control process is extremely important. As a small company, we have leveraged the strengths of our partners who have already registered with the FDA and they have a great clinical program. And it's a third party because then the validation of our cells is done completely devoid of any kind of company bias. And these third-party providers are able to give us a certificate of analysis that can show us that our product manufacturing passes the release criteria, but also the batch-to-batch manufacturing is within 10% standard deviation to each other, such that one patient can get products that are manufactured from different campaigns and vice versa. So really, the scaling of our technology has been not easy, but it's really amazing where it stands right now. In addition to back to the commercialization side, on the commercialization side, we are creating redundancies. We are talking to CDMOs. We are engaging with the thought of, building our own facility, but we have now standardized our potency assays. We have leveraged the knowledge that's out there with other companies that have done so well. And we are trying to make sure that when we are filing for our BLA and we are going for the next step, we are able to check all those boxes that are and follow the FDA guidance.
Yeah, sure. Yeah, thank you. A couple more questions for you. How do you really envision Cellenkos' Treg cell therapies, either complementing or competing with existing treatments for both autoimmune and inflammatory diseases?
Thanks, Erin. So that's actually a great question because I think that what we are creating is a paradigm shift. We are really transforming the disease from inside out. And the trials that I have been speaking about, we don't prescribe the doctor to change their underlying therapy. And most of our treatments have been an add-on to their existing lines of therapy. So we feel that our cells can be a great complement to existing therapies as well as novel therapies because of non-overlapping mechanisms of action and also not creating any interference in the actual target. So, going back to the CK-804 data, we had treated patients who were already on a stable dose of ruxolitinib, which is a JAK inhibitor. And when we added the Treg cells, it actually didn't make any changes in their potency, but it actually relieved them of the side effects. We have seen that in our preclinical model also, that when we are giving our cells to the mice who have been treated with ruxolitinib for treatment of GVHD or lupus, we actually relieve the inflammatory component. So we feel that our cells can be really added on to any other therapy out there. In patients with aplastic anemia, they continued their cyclosporin and eltrombopag, and our cells were layered on top of it, and they synergized with the existing therapies to create these long-standing transfusion independence. The other forum that we see ourselves to be implemented is as a single agent therapy in diseases where no treatment exists. So, I mean, this is very exciting. It's preclinical right now, but we have created a genetically modified cell that can be used in babies with IPEX syndrome that don't have FOXP3 gene and basically succumbed to Tregs or autoimmune diseases and massive inflammation. But last but not the least, in terms of competing. So I feel the biggest, and right now I'm going back to my days of transplants. I think that one of the near-term displacement that we see is that in patients with aplastic anemia, where it's a benign disease and they require allogeneic stem cell transplant, if we are able to create these kind of transformations from inside out, and if we are able maybe someday to infuse our cells early on, we may be able to displace these otherwise onerous therapies. But, it's a lofty goal and we hope we will reach it. But that's where we feel that we can maybe displace or maybe compete with some of these existing therapies that are so difficult for the patients to tolerate, and they carry really high risk, including their own mortality risk.
Sure. And perhaps that is even part of your response to my next question, but still want to talk through it anyway. What would you say, what's next on the 2025 roadmap for Cellenkos?
So in 2025, we are now converging to be really focused on getting our lead product to market. So you can see the story of our company. We started in GVHD in the academic setting. When we went into the company, we didn't go after GVHD for two main reasons. One was at the time that we started, we didn't have the cryopreservation and off-the-shelf manufacturing streamlined. And number two was that we wanted to go after not a man-made disease, but a disease that exists in nature because of the very mechanisms of action that I had described earlier, so that we are able to show that a patient is diseased, we give our cells as treatment, and there is an effect size that can be measured and attributed to the intervention that was made. So as a result, in the past 6-7 years, we have gone after patients with aplastic anemia, with myelofibrosis. We have treated 12 patients with neuroinflammatory disease, including amyotrophic lateral sclerosis, ALS. We have some very exciting data coming out in publications. And during COVID, we treated patients in a placebo-controlled trial with lung inflammation. But with all that enriched experience in the past, we are now converging the safety data and the mechanism of action and our own understanding of how these cells work as a therapeutic. And we are putting all that information and intelligence behind our lead product, which is CK-0801. So in year 2025, Cellenkos will be pushing forward with a registration trial for CK-0801 in patients with aplastic anemia, who have either a suboptimal response or no response to their prior therapies, such that we can transform their disease and hopefully lead to not only independent from blood and platelet transfusions, but with great quality of life and a decrease in burden on healthcare, decrease in their own absence from work or absence from being with their families, because these patients are so burdened by just making trips to the hospital. Mm-hmm. So our goal in 2025 would be to put CK-0801 on the map, to get it through the approval process, and hopefully make a difference in many, many, many patients' lives.
Wonderful. We'll be following you along as you do make those strides for sure. And all right, listeners, that's it for this episode featuring Cellenkos' Dr. Simrit Parmar. Simrit, thank you so much for your time and the insight you shared today. This was marvelous.
Thank you, Erin. Thank you for your time.
Absolutely. Thanks for tuning in. Be sure to visit Cellenkos at cellenkosinc.com and bookmark Cellengene at cellengene.com for the timely content that could help you perform your role even better. And we'll talk to you soon.